Okay, welcome to our Thinking Beyond. Before we get started with tonight's talk, I just wanted to announce that our next Thinking Beyond is on April 29th, and we'll have guest presenter Mark Jackson, and he will be giving a talk called The Race for Quantum Computing. And I will send out the registration links for that mid-April. And then just like usual, if there's any questions during tonight's talk, just use the Q&A feature at the bottom of your Zoom screen, and we will get to those at the end of the hour. And so that's all I have. So I will turn it over to you, Paul, who will be introducing our speaker tonight.
Well, yes, thank you, Jessica. It's a pleasure to introduce my old friend, John Mattick from Australia. I first came across John when he was at the University of Queensland and had created the Institute for Molecular Bioscience and the Australian Genome Research Facility at the University of Queensland. And I seem to remember him saying that when they started, there were only 4 people. And I think then at that time there were like 400. So that was a huge success. He's had a number of other positions, including in the UK. I think of him also as the director of the Garvan Institute of Medical Research in Sydney. And his current position is at the University of New South Wales, which is also in Sydney. He's the Sharp Professor of RNA Biology there. He's had many awards over the years, and he says not to dwell too much on it, but he did want me to mention the Hugo Award. And Hugo, that means, let me see if I can get this right. This is, I'm struggling to see where it is. Here we go. So this is the Human Genome Organization Chen Medal, which was part of this, for distinguished academic achievement in the human genetics and genomic research. So I'm just reading this from the screen. I probably made a hash of it. So John, I'm going to hand over to you. You correct my mistakes, and if I've left anything important out, now is your chance to tell us.
Thank you, Paul. That was good enough. That's fine. Hello, everybody. Thanks for joining, and thanks for the invitation. It's really great to be with you. It's I'm finding this whole Zoom thing, terrific, because otherwise, I'd have to travel across the Pacific, and that's got its own advantages. But I'm really glad to be able to reach out to different people by this method approach. So appreciate the invitation, Paul and colleagues. So we're going to go right into it. I'll just start sharing my screen. Let me know if you can't hear me or anything else is wrong. There we go. Okay. So hopefully that's come up okay. There's a title slide. Look at the theme of this talk is suggested by Paul as the dark matter in our genome, which was a term coined some years ago by a science journalist for Science magazine. It's getting a little dated now, I think, as people realize that was perhaps a term that was coined when we didn't know much about what was happening. But the fundamental theme of this talk is that the field of molecular biology is pretty fundamentally misunderstood the structure of genetic programming of complex organisms, including especially humans, because of the assumption made decades ago in the 60s and 70s that most genes encode proteins, which is largely true for bacteria, but turns out to be untrue for complex organisms, and so untrue as to be misleading. I always like to start these talks with a picture of one of my favorite scientists, Barbara McClintock, and you'll no doubt have heard of her. She got the Nobel Prize for discovering transposition or jumping genes in the 1980s. And this is a photograph from the front cover of a book written about her. There's a picture of her as a graduate student in front of her beloved Maves at the University of Missouri. And she was one of the great intellects of the 20th century in genetics, and was much revered, contrary to some popular opinion. She was elected as president of the American Society of Genetics in 1948. But her key message was kind of not understood. And I want to just show you a quote from this book and from a letter that she wrote to a good friend, Marcus Rhodes, in 1950. This is 3 years before the double helix. And Barbara writes, are we letting a philosophy of the gene, that is, as a protein coding entity, control our reasoning? What then is the philosophy, and is it valid? And she goes on to say, when one starts to question the reasoning behind the origin of the present notion of the gene, held by most geneticists, and she should perhaps also have said biochemists, because the idea was that genes made enzymes, one gene, one enzyme, which morphed later into one gene, one protein. But she says the opportunity for questioning its validity becomes apparent. Now, what she was intuiting here, in the best sense of that word, was that genes were more than just a specification of the component parts, the proteins or the enzymes of an organism, that they also were control elements, because she had seen under her microscope in cytogenetics bits of DNA jumping around that changed the phenotype of the corn, of the maize. And she kind of intuited correctly that these jumping genes were not encoding enzymes, they were control elements. And she insisted on that throughout her life. In fact, she became the Grand Dame of Cold Spring Harbor, but went to her grave frustrated because while she got the Nobel Prize for discovering the phenomenon, its implications were really not appreciated about the fact that they were control elements for development. So as I said, the assumption based on the studies of the lac operon, and those of you who have done any biochemistry will be familiar with this, in the enteric bacterium Escherichia coli, has been that genes are generally synonymous with proteins, and that most genetic information, including regulatory information, is transacted by proteins. And this protein-centric view very deeply reflected the mechanical zeitgeist of the age. We're talking in the 50s, 60s, and 70s, and led to many subsidiary assumptions, despite a number of subsequent surprises that should have given pause for thought. The first, which is where I got interested in this, and I was at the time a postdoc at Baylor College of Medicine in Houston, is that genes in humans and other complex eukaryotes are actually mosaics of protein coding and non-coding sequences called exons and introns. In other words, in bacteria, a gene is collinear with its translated protein product, but in us, there's a bit of the gene which specifies a little bit of the beginning of the protein, then a whole lot of other nucleotides, and then down there somewhere on the other side of, Phoenix is another bit of protein coding sequence, etc. And this was, in fact, the biggest shock by far in the history of molecular biology, because nobody really anticipated that genes encoding proteins would not be collinear with their encoded protein products, with the genetic code. But the immediate and universal interpretation, all the way to Crick, was that the intervening sequences, or called introns, despite the fact that they are transcribed into RNA, but which is not translated into protein, must be junk. And of course, there was another and equally, in fact, more plausible and certainly far more interesting alternative, is that other information is being transacted by this non-protein coding RNA, which is what popped into my mind on Friday night having a beer in the pub in the Texas Medical Center. The second surprise, but at the same time, is that our genomes are full of transposon-derived sequences. These are mobile genetic elements, a la Bravo and McClintock, coming from retroviruses and other mobile sequences that can jump around the genome. And the interpretation, again, same dodgy logic, was that these sequences are mainly non-functional, selfish DNA. Books were written about this, the selfish gene. The third surprise, which came a little later with the genome projects, is the gene number in animals, or in particular, doesn't scale with developmental complexity. And there are all sorts of rationalizations about this, about combinatorial control, which I won't go into, but except to say that they were never justified theoretically, mathematically, or mechanistically. It just said, oh, yeah, that's combinatorial control, and that'll scale as a factorial, and that'll explain everything. But it was just intellectually lazy on the part of the biochemists who just wanted to keep believing that proteins did everything. And these enduring interpretations are examples of what one might call founder fallacies, often, or usually, followed by what's called validation creep, where people, initial observation is made, and naturally and properly people try to generalize from it. But if the truth of that generalization can't be tested at the time, the original interpretation becomes the orthodox explanation, and then graduates into an article of faith, and it becomes almost impossible to dismantle it until we get to anomalies, which I'll get to at the end of the talk. And that's not as well as the way through. So as I was saying to Paul before we started, most of my colleagues in molecular biology are the intellectual descendants of biochemists and geneticists. They tend to focus on a particular protein or gene or pathways, very reductionist, and never stand back, or very rarely stand back and look about the whole nature of genetic programming. So here's the third anomaly. The humans and this little worm on the right have approximately the same number of protein coding genes. Now, C. elegans is a nematode worm. It's only 1 millimeter long, has 1,000 approximately somatic cells, few nerve cells, few muscle cells, and a gut. That's it. The organism on the left here, us, has got, as an adult, about 40 trillion cells, give or take, arranged into a myriad of organs, muscles, and bones that I'll talk to in more detail in a moment. And yet they both have the same number of protein coding genes. And this was a huge shock because there was a lot of debate before the genome projects about how many genes humans might have. And there's an interesting intellectual history to all of that, including their whole idea of junk DNA or dark matter. What was worse is that most of the encoded proteins in these organisms are similar, have similar functions. And many are actually common with simple eukaryotes like yeast, the proteins involved in basic cell biology. So the question arises, how much information is required to program human development and cognitive capacity, and what form does it take? Now, let's just look at this in a bit more detail. Now, the most interesting organs, I think, in humans or mammals, are the bones, apart from the brain, of course. And here's just 3 vertebrae from the cervical, thoracic, lumbar parts of the spine in 3 different planes. And you can see how precise the architecture is here. An anatomical pathologist can look at a vertebrae in the ground and immediately say, hey, that's, L3 or C4, whatever, exactly which part of the spine it comes from. Here's a thing you might see commonly in physiotherapists, cases where you look at all of the muscles and ligaments and the beautiful architecture of these things that has to be programmed from the point of conception through very many cell fate decisions. In other words, we start out life as individuals at the point of conception, single cell, and it has to divide and differentiate into this level of specificity. Now, the full cellular ontogeny of C. elegans has actually been studied and characterized by mainly in Cambridge, England. And this is the diagram of the origin of all the different muscle, nerve cells, etc., the 1,000 cells, which have been followed under the microscope. And we know that ontogeny from C. elegans is precise and reproducible, except for genetic variations, and the same occurs in humans. And we can demonstrate that simply by looking at monozygotic twins or identical twins, that essentially complete phenocopies. So the program that we inherit from our parents, with all its idiosyncrasies, is what programs our development, to a large extent our psychology, and we can get into that perhaps in discussion. But so the difference between us as individuals is not because of any in utero effects. It's actually just the variations in subtleties in our genetic programming. And if you run the same program twice, you get the same outcome. In the case of C. elegans, 1,000 cells involves 2,000 cell fate decisions in this binary tree. In our case, we require something of the order of 80 trillion cell fate decisions. And that's 4 orders of magnitude more than linear information content of the human genome. Think about it. We've got to go through tens of trillions of cell fate decisions to put all the bones and muscles in the right places with the right architecture. So it's not just a matter of cell type, like a bone cell or a muscle cell. It's actually architecture, and it has to be done with this complete precision and reproducibility. Now, one of the clues to this, although it's superficial, and is that the Genome Project showed the extent of the non-protein coding DNA, that's introns and in protein coding genes, but also sequences between genes, which were called intergenic. The extent of that DNA increases with developmental complexity. And here's just a graph that plots the proportion of non-coding DNA, topping out at us at about 98.8% non-protein coding DNA in our genome, the dark matter. And we also knew in the following years, so the Genome Projects really came to fruition around the turn of the century. In the following half decade or so, there was a lot of RNA sequencing done at high throughput using the same technology. And what came out of that was that irrespective of the extent of these non-coding sequences, the vast majority of them are actually dynamically and differentially transcribed into RNA that's not translated into protein in different cells and tissues at different developmental stages. So you force the conclusion that either there's an enormous amount of useless transcription, or there's another layer of genetic information transacted by this RNA, which expands progressively in complex organisms. Now, the data for this, I'm going to show all of it, but this was the main one came out of Yokohama in Japan, the RIKEN project group there. And we were part of this international consortium and very privileged to be so. This was a paper published in Science in 2005. And the motivation for this work was to do high throughput RNA sequencing to try to better define the proteome and the spliceome, the isoforms, in mammals. But the surprising and ultimately headline result that came out of this work was that there were tens of thousands of transcripts being produced that had little or no protein coding potential. What's this stuff? In the accompanying paper, we showed that about 70% of mouse genes exhibit overlapping antisense transcripts. So it's not just the introns in the middle of genes being transcribed or intergenic regions, but the other side as well. And produce some preliminary evidence that this might have a regulatory function, which is the logical prediction. And even at that time, this is what, 2006, it was obvious that the mammalian transcriptome is a continuum of genetic information. This idea that genes are discrete entities is completely wrong. Genes, this is a cartoon with blue blocks here represent protein coding sequences, white blocks non-coding sequences, and we see that there are multi-exonic, that's what the blocks are, with introns between them that are not protein coding, that may be protein coding, that overlap other genes, that encode small RNAs within them. And even when we drew this diagram, almost 20 years ago, we knew it was an oversimplification of the actual situation. In fact, it was later shown by Tom Gingerison group at Cold Spring Harbor that many genes, the genes are expressed embryonically in Drosophila or in mammals, have exons, that's the first part of the coding sequence, that are hundreds of kilobases upstream and span many other genes in between. So we're talking about a continuum of genetic information coming from both strands, and in every cell and tissue, the pattern of expression, the exons, the splicing patterns, etc., are different. We also showed that the RNAs that are coming, we call them long non-coding RNAs. There's small ones as well, which have received more attention and are better understood, but most of the genome is producing what we call long non-coding RNAs. And it's a problematic term because, it has the unfortunate distinction of being defined by what they're not rather than what they are. But that's what we called them in the first place because we didn't know what they did. And if you look at the half-life of these RNAs in cells, they have similar range as protein coding RNAs. So they're stable and have dynamic half-lives, etc. They also show all of the indices of functionality and genic indices, including gene promoters, splice patterns and junctions, etc. I won't go through the list, except to talk about the last two, which is that the expressions regulated by conventional transcription factors and morphogens. They show very precise expression patterns in subcellular location. Then my favorite slide, this was published in 2008 as part of work with the Allen Brain Institute in Seattle. And here's a in situ hybridization analysis of hundreds of these strange non-coding RNAs in mouse brain. So the first panel here is the primary data. The second panel is a computer transformation, which gives you better contrast. And if you look at panel B here, and let's see if I get my mouse to work. No, I can't. But anyway, excuse me, let's go back. If you look at panel B there, there's an RNA which seems to be expressed, as far as we know, only in the dentate gyrus of the hippocampus. In panel C, one that's only in the CA1 subfield of the hippocampus. In fact, we've just knocked some of these out in mice and getting very interesting phenotypes, separating cortical and hippocampal memory, or losing spatial orientation in females but not males. And we're just in the route to publish this. But so we're starting to get some phenotypic information about these really odd RNA patterns. But if you look through in the cortex and the cerebellum, you see very precise expression patterns for these RNAs. And we looked at over 1,000 of these, and most of them show these sorts of patterns. And most of them also show very specific subcellular localization. Surprising number, by estimate, our estimate, about 30% go into the cytoplasm, and we think they're involved with formation of phase-separated domains, which I'll get to shortly. Most of them are expressed in the nucleus and controlling gene expression patterns, which we'll also talk about. Others are decorating previously unknown and still uncharacterized subnuclear organelles. Others are in paraspeckles and neurites, etc. So they're not only showing very specific expression patterns at a cellular or tissue level, but also very specific subcellular localization, which does not really sync with the idea of noise, although that was the common reaction from the field for this, because these things are very lowly expressed. But in fact, they're not lowly expressed. If you go back to the previous slide in your mind, they're very precisely expressed. But if you mash up a mouse brain and say, you'd be lucky to see that RNA in panel B, because it's only expressed in a few cells within the brain. So I'll just flick through this again. Sorry, I should have gone back. And some of these, show patterns of expression in particular types of cells more commonly. This one identified by Shinichi Nakagawa, again at Riken, which he showed constitutes a novel nuclear domain in a subset of neurons. And then we were able to show that this particular RNA is acutely downregulated in response to neuronal activation and shows the same patterns of changes in alternative splicing as we see in schizophrenia, and identifies with the same proteins, associates the same proteins that are implicated in the etiology of schizophrenia. Now, what also has come out over the last few years is that most genomic regions associated with complex traits and diseases express these long non-coding RNAs. And I don't have time to explain how this is done, except to say large-scale genotyping of indicative single nucleotide polymorphisms across large populations that show either a particular trait versus controls or a particular disease. And these are called Manhattan plots. And here's an example. So if you plot by whatever measure was used to intelligence in this case, genomic variations associated with large populations that show whatever high intelligence, and then look at the frequency of particular variations in the population that are associated or not with this, you get this these peaks that show up above random variation or background. And these are called Manhattan plots for obvious reasons. So what came out of this was that very few of these variations that affect complex traits and diseases are protein coding. Most of them are intronic and intergenic, that is regulatory. So there's regulatory variations which are affecting complex traits, and that includes psychiatric traits, physical traits, etc., and complex diseases like neuropsychiatric disorders, etc. So we looked into this in high resolution, and you got time to read this. There's an extraordinary range of things that have been polled this way. Premature ovarian failure. I'm just looking randomly through here. Hippocampal atrophy, panic disorder, you name it. And so what we did was to analyze what was coming out of these haplotype blocks associated with these disorders. And here's one example, one of my favorites. So the red bar here is the haplotype block in the genome that's been polled. In the database, at least at the time down the bottom there was nothing recorded. Here's the sentinel SNP that's used in the chips that do these analyses. And we find right in the middle of this is a multi-exonic long non-coding RNA, which has to be a prime candidate for the mechanistic basis of what was being measured in terms of cognitive performance in the study. Here's another one showing three non-coding RNAs coming out of another big block. And we found that most of these things were expressing these RNAs, albeit at low level or in particular sorts of cells. And then we went on to looking at these things in neuropsychiatric functions. I don't expect you to read this, but rather we found that nearly all of the regions associated with neuropsychiatric, neurodegenerative diseases, etc., were expressing these long non-coding RNAs. We and others, and this is our work here, but others who have done the same, have looked at the dynamic expression of these RNAs in many different contexts, including embryonic stem cell differentiation, neuronal cell differentiation, immune cell activation, muscle differentiation, breast cancer, memory development, melanoma, etc. And in fact, there are now thousands of publications reporting phenotypes from the knocking out or knocking down these RNAs in one context or another. So we now have a whole range of phenotypes in terms of development, brain development, viability, immune system, etc. This is a recent review that we wrote for Nature Reviews Molecular Cell Biology. Here's one example of a long non-coding RNA that, when it's in a heterozygous state, produces skeletal abnormalities, just to put a bit more tangibility on that diagram. They're also involved in cell fate programming and reprogramming. And here's again a review from about a decade ago, many more since then, but these RNAs have now been implicated in the, the regulation of the biology or the development, differentiation of neuronal tissues, keratinocytes, skin, heart tissue, etc. Now, some years ago, my colleagues Marcel, Paolo, and I and others looked at the dynamic expression of these things in embryonal stem cell development. And again, it's a complex diagram. But what came out of this was that these long non-coding RNAs in red here, well, these are the two we focused on, which were antisense to important protein coding genes that control development, were being dynamically expressed during embryonal stem cell differentiation. And we were able to show for the first time that these things are associated with chromatin modifying complexes and modified histones. And you can see in this diagram here, the HOXB5B6 antisense is binding to a protein called MLL1, which is a chromatin activating protein, or particular forms of modified histones are associated with active genes. So a year later, our good friend John Rinn, whom I just he's in Boulder, Colorado. We were just emailing each other this morning. He writ this large and showed that with over 3,300 of these long non-coding RNAs, he did this at the Broad Institute at the scale that only the Broad can do, showed that about 20% of these long non-coding RNAs were expressed in different cell types. They are bound by polycomb, which is another chromatin modifying complex, a repressive one. And if you knock them down, you get activation of these genes that are normally repressed by polycomb. So just to introduce those of you who've probably vaguely heard about epigenetic processes, and epigenetics generally, it's clear, and it's been known for a century, that epigenetic processes are central to differentiation development, and that's probably their most important function. In other words, they're modifying the chromatin to organize the patterns of gene expression during these trillions of cell-fate decisions we have to make with accuracy during our development. They're also, and this is where the more popular interest that comes from, they also mediate long-term responses to environmental variables like in type 2 diabetes, and big players in cancer, and they're important in brain function. If you knock out the epigenetic processes, you cannot learn. So the brain is actually evolving on the fly in response to experience using these epigenetic processes. Now, the epigenetic processes or memory is embedded in the modification of nucleotides in DNA, cytosines mainly, and in a wide range of modifications of the histones that package DNA in a nucleosome. And so our DNA is not just a linear string, it's actually wrapped up like around these balls of protein called histones. Histone, there's 4 of them in each octamer, and this is the unit of structure of the chromatin. Now, these histone modifications that occur, excuse me, I'll just go back to this. The modification of histones, they stick out the tails of these structures, are catalyzed by a suite of about 100, or in fact more, generic enzymes that impose a myriad of these different chemical marks. So there's over 100 of them, at literally millions of genomic locations in different cells at different stages of differentiation. And one of the great things that I learned while I was writing the book I'm going to refer to at the end was that in the brain, these chemical modifications include the covalent linkage of dopamine and serotonin, which are important neurotransmitters. So what's going on in our brain is also being registered in epigenetic memory. So the question is, what determines the site selectivity of these enzymes? Because they have no sequence specificity, they're just enzymes. What determines the positioning of nucleosomes? Is that important? Turns out it is. And what's the molecular basis of the epigenome environmental interactions, including in the brain during learning? And the answer to all three of these questions is RNA. So it turns out that most of the RNAs that I've been talking about, link RNAs, are coming from genetic loci called enhancers, which were recognized initially, back in Drosophila with Ed Lewis and others, a century ago or 80 years ago, but thought to be just cis-acting protein and transcription factor binding sites. But the evidence that's emerging very rapidly now is that these genetic loci are actually genes that produce long non-coding RNAs. And in fact, I'm just summarizing here in text, forgive me, but just to get through it quickly. So it turns out that enhancers are genetic, well, this has been known for a while. These are genetic loci, that is genes, that control the spatio-temporal patterns of development in plants and animals. There are estimated very roughly to be approximately 1 million, give or take 500,000 enhancers in the human genome. There's a lot. And these enhancers, like protein coding genes, express long, multi-exonic, highly alternatively spliced RNAs in cells in which they are active. Genetic knockdown studies have shown that enhancer RNAs are required for enhancer function by guiding effector proteins to target loci in what's called phase-separated transcription splicing hubs. Now, I'm going to talk about phase-separated things in a moment, but this is probably the most exciting development in cell and developmental biology in the last decade or so. That is finding that the cell is far more highly organized than we thought. Now, the production of these long non-coding RNAs from these enhancer genes explains their very cell-specific expression, their rapid evolution under positive selection for adaptive radiation, which has always been said, oh, they're not very well conserved, they can't be useful, can't be important. Their subcellular localization and the genome-wide incidence of the features of regulated expression, such as transcription start sites, transcription factor binding sites, splice sites, and epigenetic marks, which were revealed by the ENCODE projects. And the production of link RNAs from these enhancer genes resolves the G-value enigma. In other words, we don't just have 20,000 genes, we've got 1 million or so genes, but most of them don't encode proteins, they're actually encoding the RNAs which are controlling the patterns of development. So a way of thinking about this is to think about the proteins as the Lego parts, most of which are common to all animals, muscle proteins, nerve proteins, etc. And the differences between different animals and their complexity, but also their, adaptive specializations, wings or big teeth or whatever, is actually programmed by the architectural plan. So if somebody gave you a set of Lego parts and gave you a small set of plans for a house, you could build it and give you a big set of plans for a castle. Those plans would be much larger, and there might be a few extra specialized parts, but basically it's the complexity of the plans which determine the complexity and the phenotype of the organism. Now, it turns out that RNA controls this phase separation. So if I go back here to this one, these enhancer RNAs are actually acting by forming transcription hubs in the chromatin. So, and this is a review from a few years ago now in Science, showing that RNA controls this phenomenon called phase separation. Now, this is liquid physics. So we now know that most, if not all, subcellular domains, I'm not talking about the membrane-bound ones like mitochondria or Golgi, but just the unmembrane-bound domains, are actually phase-separated or liquid crystal domains that are nucleated by RNAs interacting with proteins containing these things called intrinsically disordered regions, which were enigmatic and not understood for ages, the IDRs. These domains, and people have been looking at this for over a century, include nucleoli, which you can see under the microscope in the nucleus. They're particular domains in the nucleus, but they're not membrane-bound, as well as a bunch of others that I've mentioned there. Now, these IDRs are present in and essential for the function of nearly all of the proteins involved in animal and plant development, including transcription factors, the homeotic proteins, histones, etc. And the fraction of proteins containing IDRs in the protein in organisms correlates with the number of specialized cell types. And there have been big increases in the fraction of the proteome comprised of IDRs between prokaryotes, simple eukaryotes, and multicellular eukaryotes. In fact, this is the only part of the proteome that I'm aware of which actually co-evolves with developmental complexity. And eukaryotic chromatin in our nucleus is organized into topologically associated domains or phase-separated domains, which are likely nucleated by enhancer RNAs. In other words, that's their function, is to nucleate these things and guide the relevant proteins to determine the patterns of gene expression at each stage of our development, to have very precise decisions made about divide or differentiate, divide or differentiate, divide or differentiate, trillions of times. Here's a picture of these domains at single molecule resolution, long encoding RNAs. You can see they're very specific locations and show association with these phase-separated domains. And this is a cartoon which is almost certainly wrong. And when I drew this last year, I pointed out in the paper that these things these cartoons are meant to try to visually explain the concept, but they shouldn't be taken too literally, otherwise they can be very misleading. And here's one of my favorite pictures taken by a Japanese microscopist showing that RNA binding to DNA in triplex, and they can do that through the Hookean groove and the double helix. Looks like the Milky Way. RNA is talking to DNA all over the genome. This is the nucleus. These RNAs are actually scaffolds and guides. They bind proteins because RNA is not a linear structure. It's actually a very complex folded structure, which has a great advantage of being able to form secondary, tertiary structures that can be recognized and bind to proteins, as well as having open regions that can target other RNAs or DNA. So just to sort of wind up, when we entered this field in the 1980s, nobody knew how many protein coding sequences. In blue there on the left, there was a big question before the genome projects. We knew there were lots of introns, but it was thought that most of the genome was not functional or dark matter. By 2000, with the genome projects, we had better purchase on the protein coding sequences, which were only 1 or 2% in humans. We picked up a lot of other particular RNAs that have special functions. We got more information about introns, but we thought the rest of it was still not functional junk. But now on the right, this is the emerging view, is that the whole genome is functional, and that most of the genome is producing RNAs that regulate and organize the chromatin and the cell biology, including translation. But their prime function is to organize the topological domains in our chromatin that then lead probably through the centrosome, but this is another frontier of research, into accurate four-dimensional divide or differentiate decisions that takes us from one cell to a walking, talking adult. Now, I've got there a paper I wrote this last year on this topic. You will recall that Thomas Kuhn wrote a famous book in 1962 called The Structure of Scientific Revolutions. He made 2 points. He said that basically science lurches, from, this is my term, from paradigm to paradigm, and in between, so there's a conceptual framework established, everyone believes it, and in between what happens then, you have what he called normal science, where people design their investigations and interpret their data or their results within the conceptual framework that's been established, until anomalies appear that suggests that there's something wrong with the conceptual framework. Now, these anomalies are usually ignored, as they have been in molecular biology, until they actually accumulate to a sufficient level where people say, oh, gee, we haven't got a complete understanding of what's going on here. So the first condition for a paradigm shift, according to Thomas Kuhn, was the accumulation of anomalies. But he said quite accurately that the prevailing conceptual framework will not and cannot be replaced until there's an adequate substitute that respects the data from the past, but actually puts it in a more complete, framework. And you could think about, Newtonian to Einsteinian physics, or any other of the classic paradigm shifts in the history of science. So that's what's happened here. Basically everyone thought genes made proteins, proteins did everything. But now we've got all these anomalies occurring in terms of the structure of the genome, the expression patterns, and now the functions of these RNAs, which now say, oh, no, that was incomplete, so incomplete as to be misleading or wrong, that most of the genes in humans, and we've got 1 million, give or take 500,000 of them, that actually produce RNAs that actually are the convey the architectural plans to take us from 1 cell to 40 trillion. We also know, and this is really the end, I don't have time to talk about it, but the RNA is also the substrate for the environmental epigenome environment interactions, including the brain, and this is now starting to come out by deep sequencing of synapses, that there is extensive editing, as a deamination and chemical modification of all different sorts of the RNAs, which is responding to environment. So basically, the RNAs that we produce during development are hardwired to put fingers and toes in the right places, but in the brain in particular, but also physiologically, they're soft-wired, which can then feed back into changing the epigenetic state of the cells. It's a paper written in Science some years ago now, but the bottom line is that, you know, the view for half a century was that the human genome was basically, you know, oases of protein coding sequences in a desert of junk. But we now know that actually they're islands of protein coding sequences in a sea of regulation. The work was done in these 2 places that are these lovely pictures here. They're very pretty institutes where I've worked. And of course, I've mentioned some of the people on the way through that have helped us in these studies. And I'll finish just with this because I brought all this together in a book which was published last year at the end of 22. It's open access, and it charts the whole history of molecular biology from the point of view of RNA, and with the view in mind to actually take my colleagues through the history of RNA and see where the assumptions were made and prematurely became cemented until anomalies occurred through the last few decades. So that's my spiel, Paul and colleagues, and happy to discuss and answer any questions you might have.
Excellent, thank you. We already have a few questions coming in the chat, so I'll start on those in just a minute. But I want to invite everyone in the audience to please enter questions you might have for our speaker tonight in the Q&A. That was a really engaging discussion, so I'm sure we'll have a lot more questions coming in. I'm going to start with a question from Barbara Temple, which was the first one that was posted, but it's very good leading off of your last point about Kuhnian revolutions. And so Barbara writes, you said that a reason for why people clung to the idea that if a gene isn't protein coding, it must be junk is a product of the mechanistic thought of the 1950s, 1960s, and 70s. What steps can science take to ensure that it does not become biased by whatever the fashionable thought of the age is?
That's a terrific question. Thank you, Barbara. I'll dance around this one a little bit, if I may. The idea that genes made proteins was, when I say mechanical zeitgeist, this was, you know, before the conceptual framework of many of my students, you know, that, you know, it was all bicycles and smokestacks, and bicycles were made of parts, you know, and so were humans. There was a part to carry oxygen, a part to you know, do this or that. So it was a very mechanical view of life. So it sat very comfortably with the conceptual framework more broadly, not just in biology, but that, you know, at the time. But in fact, the whole idea of junk DNA has a deeper intellectual history, which goes back to the 1930s and theoretical biologists who made some outrageous ex cathedra statements, including that Lamarckian evolution doesn't occur, which it clearly does, and I can talk about that. That mutation is random, which is clearly not. And that's not just saying, I'm not saying it's prescient. It's not prescient, but it's not random. And I was going to say to Paul earlier, there's a fantastic book called The Parameterized Complexity by Mike Fellows, who's just returned to Australia, but he may well get a Nobel Prize for this, where he showed that in complex systems, you can't search them randomly if they're too complex, because the numbers of variables and combinations too large. So you have to develop strategies to search that space more efficiently. In other words, to search the subspace. And that's been quite influential, at least in me and others, in terms of thinking about how complex systems evolve. The theoretical biologists did get one thing right. They said that if the mutation frequency in human genes was the same as in E. coli and bacteria, we wouldn't survive, because if our whole genome was full of protein coding genes, well, then they estimated somewhere between 1 and 7 million, and they mutated at the same frequency as genes in E. coli, we would be, have dysfunctional genes all over the place, couldn't survive. It's called the mutational load problem. So on that basis, they said most of the genome can't code for proteins, or the mutation frequency is different. So they were right. In fact, they had the most accurate estimate of the number of protein coding genes of all. They estimated 30,000, which was pretty close. The mistake they made was to assume that what wasn't protein coding would be junk, and that variations in regulatory sequences, which they didn't consider, but are actually not lethal. They actually just give you variations. Now, they can be lethal if there's big deletions or particular changes, but by and large, differences between us are due to variations in those sequences, whereas variation in protein coding sequences are usually catastrophic. So it's probably a bit too long, but that's the intellectual history of junk DNA.
I think I'm going to let Paul ask the next question, because I know he has a few burning ones, and then I'll go back to the questions in our chat. Go ahead, Paul.
Yeah, well, it's great to see you waving the flag for RNA, John. And all this regulatory stuff, all these different RNAs, makes me wonder with inheritance, because back in the old days, we were told it's all down to the genes, and the genes are in the DNA, and that's the only thing that gets copied. And now you're raising the prospect of what we might call epigenetic inheritance, that all of this other stuff has to somehow be conveyed to the next generation. Now, have I got that right? And is it understood how that happens?
The short answer is yes, and partly. So the history of epigenetic transgenerational epigenetic inheritance actually goes back to 1915 to an English geneticist called Bateson, who recorded what he called rogue patterns of inheritance that deviated from Mendelian ratios. It sort of lay there until the 1970s, when Robert Brink came along and formalized this, and he termed it, and it is now known as paramutation, and it was studied intensively in plants where you can do the genetics. And there's also who's at the University of Arizona who's famous in this field? Oh dear, I'm having a senior moment. One of your colleagues is a woman. I'll look it up in a minute, but it's terrible, isn't it? She's written beautiful reviews and studied this. So there's now no doubt that transgenerational epigenetic inheritance occurs in plants, and that it's RNA mediated. There's actually very good evidence that it also occurs in animals. Interestingly, studies from tomatoes show that microsatellite sequences, same sort of sequences, are used for DNA fingerprinting. These, sort of dinucleotide and trinucleotide repeat sequences are actually the substrates for this transgenerational epigenetic inheritance. These are the ones that expand or contract in neurodegenerative and neuropsychiatric disorders. And there's about 1 million of them in the human genome. So nobody's sure the extent of transgenerational epigenetic inheritance in humans. We do know it happens in mammals to some extent. And it probably accounts for the missing genetic variation in the genome-wide association studies, but it's a new frontier. So we're inheriting not just the hardwired stuff, but soft-wired stuff that's coming. And the extent range of that is really not understood.
Which raises the next question. So one of the things we're interested in the Beyond Center is, the whole history, the origin of life, and its evolution over long periods of time. And we, of course, have the time tree of life in genetic information. You can reconstruct a phylogenetic tree, get back, and if you give me your favorite gene, we can figure out, how old it is in some sense. What about all this other stuff? Is there a time tree for the RNA regulatory mechanisms as well? Could you, from current sequencing, reconstruct a natural history of that and say that this type of control arose 2.5 billion years ago and this type 1 billion years ago and so on? Is there a whole subject there waiting to come up?
Well, I don't think so. For the reason these regulatory sequences evolve very rapidly. In fact, the Evo-Devo people will tell you that it's well established that phenotypic variation, adaptive variation between individual species, etc., is mainly regulatory variation that changes the patterns of gene expression, the size of your muscles or whatever it might be. So it evolves very quickly and very difficult to track back. You can see some of them going back through the mammals, occasionally through the vertebrates. So I think it's very difficult. The other thing is that You know, the great strength of RNA is its ability to bridge the digital and analog worlds. It can fold up, it can bind proteins, but it also has sequence specificity, so it's guide molecule. And you know, that wasn't really of much use in unicellular organisms, which dominated the planet for 2 or 3 billion years. It's a bit like saying you don't need digital controls on a bicycle. But when life was climbing this mountain called you know, multicellularity, that strength of RNA came back to the fore. So you see the evolution of these RNA regulatory networks really track with the evolution of complex organisms.
Now, there's a lot more I could ask, but I'm not going to do it because I'm looking at the clock, and we've got 8 questions per
okay, sorry. Well, I'll try and be bulletproof, but there's so much to talk about here.
Well, we can talk offline sometime, but let me be quiet, and we'll take some more questions. So, Sarah?
Yeah, excellent. Thank you. There's a couple questions from Charlie Langweaver, so I'm going to bundle 2 of them together because I don't think we'll have time to get through all of them. The first one is, you talk about animals and plants, but do you really mean animals, plants, and fungi, which I think is getting at whether fungi are...
Yeah, I do. It's
yeah.
fungi are not as complex as plants, but you know, yeah, I include fungi in there. And fungi are actually more closely related to animals than they are to plants. It's
right.
And they have a common origin as scavengers.
So are they more on the spectrum closer to what animals do with RNA than what plants do also?
No, plants are more developmentally complex, but the plants are different. They, in us, you know, it matters where our fingers and toes are. They have to be really precisely placed for our, you know, success, and not for our viability. We can survive. You know, we live with a non-functional hand, but we're not going to survive, you know, predation or whatever. So there's difference between viability and fitness. Plants are, you know, sort of stuck where they're stuck, and they have much more developmentally plastic because they have to work out which way their branches and leaves to get the optimal sun, etc. So they use transposable elements far more flexibly than we do. But they do have these enhancers that control the patterns of floral and, you know, leaf development, etc. So, but animals, particularly the mammals, are the most complex of all in a developmental sense, because we have all these myriad of muscles and bones and so forth. So, but fungi are in there, yeah. I just, you know,
yeah.
And I think I avoid fungi because, you know, so many people are familiar with the unicellular fungi like Saccharomyces cerevisiae that I don't want to have to say multicellular fungi, you know.
Right.
Whereas plants, they usually associate with multicellular plants as opposed to algae.
Right.
Like technically, you know, yeah.
Yeah. Thank you. So Charlie's other question here is, if you could say more about the function of phase-separated regions.
Yeah, this is extraordinary. It turns out, and I'm not the best person to explain it, but it turns out that in liquids, if RNAs and proteins interact, they can create an aqueous environment inside this micelle, you might call it, that's different from the aqueous environment outside, even though it's still aqueous. And that these things are then formed as specialized organelles, or want a better word, where specialized functions take place. They could be the chromatin organizing particular patterns of gene expression, but we also, you know, we think about protein translation and ribosomes. They occur within phase-separated domains. It's like the whole cell organized extremely finely by these interactions. It's like New York City, you know, you see it from space, you think it's a mess, but when you get out close, you realize that it is pretty finely organized. And we're just starting to realize that the cell is not a diffusible gel. It's actually extremely highly organized. And the rules of how the RNAs interact with the proteins and where they go is just not well understood. And all of these IDRs that I talked about, which are essential for formation of these phase-separated domains, they're very particular sorts of sequences, including positive charges that interact with the negative charges in RNA. But they're also the sites of post-translational modification. So the interactions between the RNAs and these proteins can be modified by post-translational modifications, the chemical modifications of these tails, which gives you extraordinary complexity. So it's just a new frontier. In fact, I tell my students that I used to think the 20th century was the, second half was the time of biology with all the cloning revolution, sequencing and so forth. Now, it was just a warm-up. This is the century of biology and medicine. We're just starting to scratch the surface.
So we have a question here from Perry Marshall, who laments about deep disappointment that highly publicized scientists have said for decades, and some are still saying that 90% plus of the genome is junk. Can you comment on the extent to which this belief has inhibited the progress of medicine and solutions to human illnesses? Can you name specific diseases that a better understanding of non-coding DNA will help us treat?
Yeah, so it's a really good question. They're all good questions, and the answer is kind of yes and no. The no side of it is that, the easiest diseases to track were the monogenic, so-called Mendelian disorders like cystic fibrosis, thalassemia, etc. And there's really catastrophic component damage of proteins. If you don't have a chloride ion transporter, you're in big trouble. You've got cystic fibrosis. That's if you survive at all. So that was really pioneered through the 90s and the first decade of this century. And that was a big mountain to climb. It was a really big deal to track down the gene that was causing cystic fibrosis. And they're called the rare disorders. They're kind of rare, but collectively they account for about maybe 3% of all births. So kids are affected by a protein coding mutation that is quite nasty. So, and most of the preconception screening and the exome sequencing is now done by medical insurers, etc., is targeted at that. What's just opening up, the second half of the question, is the realization that the genetic variations that underpin the complex diseases, and that's the autoimmune diseases, arthritis, cancer, well, cancer is a mix, neurodegenerative disorders, etc., are all in the non-coding regions. And they're almost certainly being transduced by variations in long non-coding RNAs. And that's just beginning to become accepted. So people are now starting to look into the disease etiology of these things, not just mapping the location of the genome that might be affecting these complex traits and disorders, but trying to get down to what the mechanism of these fragilities or variations is. But the one comment I'd also make about this is that you will not get, and there'll be exceptions, but I don't know of any, you will not get a complex disease unless you have a genetic predisposition. But whether you get it or not is dependent on an environmental trigger. So, so there's a whole new world of understanding the etiology and ultimately the mechanism, and then to therapies for complex diseases, which actually occupy the vast bulk of the disease burden of our population.
So our next question comes from Karen Hastings, which is, what is known about how diet and exercise or lack thereof affect non-coding RNA and its effects on longevity, disease, etc.?
Not much is the short answer, but one of the intriguing things that I found was that if you exercise a lot, and you just come back from a run, Sarah, you told me, your histones are modified by lactate, not lactate, what's the it's one of the keto acids that are produced by glycolysis. Sorry, I'm being silly now, but one of the metabolites that builds up in your blood when you exercise fiercely is actually then covalently modifying your histone. So there's an epigenetic memory of your exercise regime.
Wow, that's so cool.
Yeah, I'm not sure that there's a lot of evidence that heavy exercise extends life other than to keep people reasonably healthy as opposed to obese. If you're obese, you've got problems, but you maintain a relatively healthy weight. I think genetics are more important than anything else, provided you don't abuse yourself by smoking or getting too overweight.
I see. Excellent. Thank you. I think we're out of time for questions. Unless, Paul, you had one more burning one you want to, we're actually out officially out of time. I thought we had 1 minute, but we're out of time. So I think we'll have to save our questions. But thank you so much. That was really wonderful. I want to have everyone thank our wonderful guest, John Mattick, again, for enlightening us, and for a few questions in the chat about whether the recording would be available. It would be posted on YouTube for people to watch if they want to catch all the papers and things that were shown. So, so thank you again.
Thank you, Sarah. Thank you, Paul. Thank you, colleagues. Thanks, Jessica, for organising. And I'm very happy to chat offline by email or whatever. So you just got to look me up, my name at the University of New South Wales, and the email address should pop up. And I hope you've enjoyed it. And I certainly have had fun, telling you all about this new view of genetic programming.
Very exciting. Thank you so much.
Thank you, John. I will say good night to all our folks in the Northern Hemisphere, and good morning to those, good afternoon to those in.
Yeah, it's 1 o'clock in the afternoon. It's lunchtime. So
Dinner time for us. All right. Bye, everyone. Thank you.
thanks a lot, everybody.